Welcome everyone, and thank you for joining this live webinar. My name is Marjolein Althoff, and I will be hosting today's webinar. Today we will be going over CCI testing strategies and the proposed revisions to EU Annex 1, since this will likely have a significant impact on your CCI testing practices. I am joined by our panelist member, Josine Wilmer, Senior Study Manager at Lighthouse, and we will be available during this webinar to answer all your questions. During the webinar, you can post your questions by using the Q&A window. As shown here, please do not use the chat function since we will only be using the Q&A function to answer the questions. Also, at the end of the presentation, we will try to answer as many of your questions as we can. We will also email you a link to the PDF slides and webinar recording after the webinar so you can review the slides again at a later moment. Then I would now like to introduce to you formally our speaker, Dr. Derek Duncan. Dr. Duncan began his career at the Dutch Institute for Atomic and Molecular Physics in Amsterdam. He then moved into industry holding product and application development positions. And currently at Lighthouse since 2003, Dr. Duncan is responsible for developing applications for process monitoring and finished product inspection. With that, I encourage Derek to start his presentation, and I hope everyone will enjoy this webinar.
Thank you, Marjolein, and thank you especially to all the attendees who have logged in for this webinar. We tried to pick a time that would span several world time zones. We can see that people have dialed in from or logged in from the US, from Europe, and from Asia. So good morning, perhaps a very early morning for you dialing in from North America, and a good evening for those of you in Asia. Good afternoon. To those in Europe, I am based in Amsterdam, as Marjolein said, and over the past couple years have been following the proposed revisions to the EU Annex 1, which is going to be the topic of our webinar today, in particular related to CCI testing. So I hope to do that basically in two main sections for the next 50 minutes or so. In the first half of the presentation, we'll focus on the regulatory environment for CCI. Obviously, Annex 1 is our guidelines, manufacturing guidelines from the EMA, and will be a new regulatory guidance being implemented soon in a new version. And for the second half of the presentation, what I will do is walk through a few case studies which basically demonstrate or describe some possible CCIT solutions which could meet the proposed Annex 1 requirements in the area of container closure integrity testing. And you can see some of these case studies identified here: filling line CCI qualification, statistical CCI process study, and raised stopper height qualification. Again, we hope to get done with the presentation itself in 40 to 45 minutes, which would leave a few minutes for a Q&A session. I thought it might be useful to start with some current regulatory guidance as a little bit of context. And what you see now on the screen are some statements from the current FDA guidance for industry, good manufacturing practice. And specifically, these statements relate to CCI, and this is what it says. A container closure system that permits penetration of microorganisms is unsuitable for a sterile product. Any damaged or defective units should be detected and removed during inspection of the final sealed product. If damage that is not readily detected leads to a loss of container closure integrity, improved procedures should be rapidly implemented to prevent and detect such defects. Now, here's another current guidance, this time from the WHO, and specifically for vaccines. And this is what this guidance says here. Containers of freeze-dried vaccine should be hermetically sealed under vacuum or after filling with pure, dry, oxygen-free nitrogen or any other gas not deleterious to the vaccine. All containers sealed under vacuum should be tested for leaks, and all defective containers should be discarded. Now, if you just take these current guidances at face value, then it really looks like CCI is an issue that should always be detected and always be removed from final product. And I think a lot of people will use their visual inspection to justify or argue that this is being done in the process. And now we come to the current Annex 1 guidance. So this is not the proposed revision, but these are the paragraphs in the current Annex 1 guidance that are related to CCI, or container closure integrity. And if you look at these statements, these first two are actually the main ones that say something about CCI testing and how containers should be closed. There's a definition for what a closed container is. There's a few sentences here saying how CCI should be tested for. And then you move into some other paragraphs which are related to CCI for capping and crimping. And actually, these paragraphs caused quite a lot of discussion during the last minor revision of the Annex 1 5-6 years ago, and ended up having quite an impact on at least the capping and crimping process in industry. So this has to do with the sealing of containers and the risk to CCI. And lastly, there's a statement which talks about maintaining particular headspace conditions. So I'll leave this for you to read through. If you have not actually looked at the current Annex 1, this is there for your reference. And this is the starting point for our discussion today, our presentation today. So what is happening right now in the regulatory area with regards to CCI? And I'll start off with the first bullet point here, which is a new USP chapter 1207 titled Package Integrity Evaluation, Sterile Products. This is a chapter, it's above 1,000, which means in the USP framework, it is not required. Everything above 1,000 are recommended guidelines. But this chapter 1207 was implemented in August of 2016, so not so long ago. In fact, a little over 2 years ago. The old chapter was about 2 pages, and this new chapter is 50 plus pages. So that gives you some indication that the regulators see a need to evolve practices in this area. And the EU Annex 1 revision, the draft version is available. That's what we'll talk about today. I was just at an FDA conference last week, and there was an update on the status of Annex 1 given by an EMA representative. So the public comments have been read. There were over 6,000 public comments that were turned in March, and the draft is currently being edited. It's hard to say when this will become final because of the Brexit and the move that EMA is making from London to here in Amsterdam as of October 1st. So that was as of yesterday. The EMA has suspended all activities except for mission critical activities to give them and their staff a little bit of time to transition and move to Amsterdam, where they will be located as of March of next year. So I don't know if they see this Annex 1 as mission critical, but we'll see if we get a final version upcoming. In any case, the current regulatory environment indicates that regulators are paying more attention to the proper design of robust CCI studies, the validation of CCIT methods, and the generation of statistical CCI data, which we'll discuss further. Let's just look at a few of the main points coming out of the new USP chapter, because again, I think that global regulatory bodies are really trying to harmonize. And so these points will probably be carried over into EU thinking as well. First of all, there's a preference for deterministic or analytical CCIT methods over some of the current what they call probabilistic methods. So here you can think about the blue dye or microbial ingress testing. And there's a recommendation that CCI testing is performed throughout the entire product life cycle as part of a coherent container closure strategy. So really understanding CCI back in the development phase. Another main point here is that the USP 1207 eliminates the requirement to compare quote unquote new deterministic methods to microbial ingress testing. So in their view, science-based data is sufficient to validate the method. And if you look out in industry and you're wondering, is anybody else worried about CCI or having to evaluate or assess how CCI testing is implemented? You are definitely not alone. Here are some recent industry white papers. The first one from the BPOG, or the Bioforum Operations Group, an industry consortium involved in the manufacture of biologics. A second reference there is a group of European peers from global pharmaceutical companies, both on the topic of CCI testing. And the point here is that the industry really deems the CCI control strategy topic important enough to discuss best practices with each other. And why is that? From what we see, we think that the industry is a little bit at an inflection point with respect to best practices in CCI. With new regulation coming out, I think there's a period of uncertainty into what exactly people should be doing in the area of CCI testing. And we'll discuss that a little more in this presentation. Now, let's do a short history of how CCI has been seen in the pharmaceutical industry. So a critical leak was always linked to sterility. So historically, leak defects have been described by physical dimensions because an easy comparison, supposedly, can be made to the physical size of microorganisms. But if you look into this in more detail, there's a number of things which make this complicated fairly quickly. So first of all, microorganisms will penetrate a defect either by motility, moving on their own, or some sort of pressure differential which drives that. And if it's motility, then liquid is required over the defect pathway, which in a real life defect could be a very complex geometry. And even then, liquid presence does not guarantee microbial ingress. It is a probabilistic event. So you could do a microbial ingress test, not see penetration of microorganisms, even in a container that had a CCI defect, because for that particular test, if it's probabilistic, it might not have happened. And if you look at the published literature, this reflects an ongoing discussion about the leak, quote, size at which sterility can be jeopardized. And so there are a number of references that are listed there, and you can see that there's statements or findings that are quite varied. And so in the end, if you look at this carefully, you come to the conclusion that characterizing leaks in terms of defect size and linking that to the probability of microbial ingress is not straightforward. So let's try and look at a broader definition of what a critical leak is. And as I mentioned before, historically, leak defects have been linked to a product sterility risk. And of course, for sterile product, this is really the basic requirement for good CCI. It has to protect the product from contamination, microbial contamination. However, there are more and more products where there is a requirement to maintain headspace gas content and or vacuum, and this is done for product stability reasons. For vacuum is often used to help the preparation of the product, reconstitution of freeze-dried product, for example. And so when you ask the question, what is a critical leak, then the base answer would be, well, a critical leak is one that would pose risk for microbial ingress and a loss of sterility. However, if you have a formulation which requires critical headspace gases, then your one of your main concerns is also going to be a loss of stability and product efficacy. So if a product is oxygen sensitive and oxygen ingresses into a vial, that will be a problem for the product. And finally, as we've discussed, a loss of headspace vacuum could result in affecting the product accessibility at administration. And so for these last two cases, you could also make the argument that in general, a container that does not allow for passage of gas molecules will also not allow for the passage of microorganisms. So the sterility requirement in some cases might actually be more basic than a requirement to maintain gas content in a particular package. Okay, so hopefully these initial slides set a little bit of the context then for the discussion about container closure integrity and what the regulator is saying about it. And so now what we will do is just dive straight into the language of the proposed draft or the proposed revisions to the EU Annex 1. And the main paragraph that addresses container closure integrity is this one here that you see on your screen. The bold words are my bold, and I've chosen those because in my interpretation, the bold words are what is new in this proposed revised version compared to the current version. And at first glance, if you read through this paragraph, the changes might seem subtle when you compare it to the current version. But actually, looking at new carefully, if you look at it carefully, the new language actually may have some significant impact on current CCIT practices. And you can deconstruct this paragraph and look at what each statement or each phrase says. And so I've done that here, and in particular with the new language that is in this proposed revision. In this paragraph, it mentions utilizing validated methods. And just to comment on this, the legacy CCI tests, so blue dye or microbial ingress testing, they're not analytical necessarily. They're difficult to validate. You can read that in USP 1207. Again, I was at this FDA conference, and there were some FDA representatives presenting on container closure integrity. And, you know, blue dye and microbial ingress testing is still an accepted CCI method. However, the message from these FDA representatives is that they are being more strict in their review of these methods. So, for example, have you really validated this method to be able to detect a critical leak for your product? And as an example, in the presentation on the blue dye test, mentioned the use of spectrometers to detect the color change and not relying on operators. This proposed revision to Annex 1 has language about QRM and some risk management. If you look at the current CCI data generation, it's fairly limited and it's mostly aimed at verification. It's not really aimed at quantifying risk and characterizing the process in terms of CCI. The frequency of testing should be based on knowledge and expertise of container and closure systems being used. So again, the emphasis is on building up knowledge. It's also in a product lifecycle approach. You see that also described in USP 1207. And here's an interesting new statement. A statistically valid sampling plan should be utilized. And from what we can see, very few people are actually sampling CCI statistically. And again, this goes back to the legacy methods that are being used. Those methods simply do not enable this. So, for example, a blue dye or microbial ingress testing, will typically involve tens of samples. If you're from the visual inspection area, then you know or are familiar with AQL approaches. And for typical batches of pharmaceutical product, an AQL framework would typically mean testing hundreds, if not sometimes thousands of samples. And finally, another interesting statement in this proposed revision: it should be noted that visual inspection alone is not considered as an acceptable integrity test method. So you go back to some of the first slides where it was clear that current guidance seems to indicate testing all containers and taking out defective ones. Well, for CCI, what the proposed revision to Annex 1 is saying is that your visual inspection process, which is 100%, that alone is not considered as an acceptable integrity test method. And I think these last two bullet points on this slide are the ones that might drive or might impact current practices most heavily. So going on to the next section, which in this revision or proposed revision, which says something about CCI. Again, very similar to the current Annex 1, where containers sealed under vacuum should be tested for maintenance of this vacuum. And what they've added here is during shelf life, also during shelf life. And I think that was an obvious, or should be obvious, but the EMA representatives in our discussions over the past few years said they put it in there because people weren't doing it, and they just wanted to make very, very clear that this type of testing should be part of your stability program. And again, this phrase, after an appropriate predetermined period and during shelf life, this statement assumes data generation that is aimed at building knowledge of product, packaging, and process. It also implies a product life cycle approach. So the Annex 1 is our guidelines for GMP manufacturing, but manufacturing decisions need to be supported by data generation that happens throughout the product life cycle. Going on to the next statement, which is completely new when you compare it to the current Annex 1, and this one states that the container closure integrity validation should take into consideration any transportation or shipping requirements. Again, it seems like an obvious statement, and I know that people in the industry do this. However, again, the word from the EMA representatives is that they specifically put it in here because they were seeing cases where people were not doing this. And if you look at this statement, the container closure integrity validation. So the assumption here is that there is robust CCI validation data that has been generated and is on hand. And taking into consideration the transportation or shipping requirements, I think air pressure studies for product that needs to be flown, or vibration studies to mimic transport, these types of studies are quite well known. There are also special situations which it's become clear pose risk to CCI. And you can think here of frozen shipments or deep cold storage. The new class of products that are coming out, cell and gene therapy, actually require storage temperatures at cryo, very, very cold. You have viral vaccines that are regularly stored at -80 and -80 freezers or shipped on dry ice. It's been shown that these low temperatures definitely introduce risk of CCI to the package. And again, you need to have data which show that your transport and shipping is not introducing that risk. And so again, the emphasis is on science-based understanding of your product and process as regards to the container closure integrity. And finally, a last paragraph here, which again has to do with the sealing of the vial and the capping and crimping process. So it doesn't say something directly about CCI, but there are some implications here. And I mentioned before that revisions to this section 5-6 years ago had quite a big impact on the industry with regards to the grade A air that needed to be present for the capping process. They've added more language here in bold, and I'll read that here. Appropriately validated automated methods for stopper height detection should be in place. Microbial ingress studies or alternative methods should be utilized to determine the acceptable stopper height displacement. Now, these raised stopper height sensors have become relatively routine because this type of language was also in the minor revision 5-6 years ago. So I think just about every capping and crimping line that is delivered today will have some sort of raised stopper height sensor on it. But what's interesting here is that very few people validate the process, and the typical response that you get when you ask someone why are you setting your raised stopper height reject limit at that particular number, well, everyone sets the limit at 1 millimeter, and that's what I think a lot of the capping and crimping machine suppliers recommend, but there's no study behind that, or there's no actual qualification or validation. So that's what this new language addresses in this proposed revision. And the implication is that the raised stopper height should be correlated to the loss of CCI. Why do you want to reject a raised stopper? One of the primary reasons, of course, is that it could introduce a risk to CCI. And that's, I think, what the regulator is looking for here. A comment on this statement. We had some discussions with the regulator last year about using microbial ingress studies, and I think everyone came to the unanimous conclusion that this is actually not a good idea. I mean, this test is just too unreliable and is not something that you will want to be doing in your production area, so I think that statement is going to come out of the final version. So the statement that says microbial ingress studies, and I think they're going to go with alternative methods, or some method should be utilized to determine the acceptable stopper height displacement. Okay, so those are, again, the sections in this proposed Annex 1 revision that have to do with container closure integrity. Now, I want to go over this slide because I think it's an important one to understand how the regulator will be looking at this, and perhaps it will also help you if you are in a facility having discussions with regulators once this guidance comes out, because of all the hot topics in the revision of Annex 1, the regulators were able to come to a unanimous agreement except for container closure integrity. And so I've heard this discussion from the regulators themselves, and that's what I'll be describing here. Basically, regulators working on this revision fell into two main groups relative to container closure integrity. There was one group who really wanted to put the emphasis on process control. So the idea was we don't want people ignoring process improvement and just rely on final product inspection. And the other group wanted to put on emphasis on CCI testing per batch. So to them, it was a critical enough quality parameter that it should be part of a batch release. And so at the minimum, part of release testing, and even 100% inspection if appropriate. So unfortunately, there is this different view from the regulator. So there's not a black and white requirement that is coming out, for example, about 100% CCI inspection, which a lot of people want the answer to. So here are some questions that a production facility can consider when assessing a CCI testing strategy. And again, these are very basic questions and don't necessarily only apply to the manufacturing phase of the product life cycle, but really also applies to the full product life cycle, also back to development. So here's some questions. Is the risk or failure rate of CCI known for each product configuration? What failure rate would actually be acceptable? Can an acceptable quality limit actually be defined? Do you have CCI data that's per product? And then for the statistical CCI data, when and how will this statistical data be generated? So these will be, I think, pragmatic questions to ask once this guidance is finalized. And if you're looking at the CCI process control side of things, what I've listed here are some potential process controls that can lower the risk of CCI defects. And again, I won't go through this list. It's there for you as reference. And I think a lot of people are probably doing many of these things already. Obviously, vendor management, doing quality control of incoming incoming packaging components, qualifying your equipment, which would include worst case scenarios, and doing CCI studies, a number of things that you can do here. However, in the end, and I think this is what the regulator is getting to, statistical CCI data needs to be generated to understand if process control works and to quantify the residual risk to CCI. So if you look at this a little bit more closely, why would you want to generate statistical CCI data? Well, again, you want to understand if process control works. You want to quantify the residual risk. And if you can do that, then this statistical CCI data can be used to define and justify a CCI testing strategy. So looking at this visually, here is conceptually, in an ideal world, how this might work. So if you have a particular product package, you want to understand if this package has a failure rate. What is that baseline failure rate? And we'll show some examples of this later. And if you have this baseline failure rate, or what the risk is to failure, then you're going to implement and improve the process controls around CCI. And then to see if that has actually made an impact, you will want to quantify your residual risk. So again, after implementing these process controls, do a study to look at how CCI what the CCI failure rate is after these process controls have been implemented. And then from that, you can determine a CCI testing strategy. And so from a very pragmatic point of view, if you're thinking about how do I evolve to this framework if I'm not doing it already, or what do I need to do, facilities will need solutions on hand that enable CCI process studies to generate statistical CCI data sets. And in this conceptual visual here, of course, if you want to understand a baseline failure rate, you're going to need to do measurements and studies there. If you're going to quantify residual risk or monitor your process ongoing, then obviously you're also going to need some CCI measuring platforms for that step as well. So what would be some possible approaches to do CCI testing or CCI inspection if you started to try and move to a framework that you feel would satisfy these revised Annex 1 requirements? So the current and the draft revision of Annex 1 does identify specific products that require 100% CCI testing. So these are fused containers such as ampules, blow-fill seal process, or blow-fill seal containers. And again, why is that required? It's because that studies have shown that there is a baseline failure rate for that sealing process, which is inherent to the process. You can't control it well enough to have 100% good CCI. And in fact, the baseline failure rate is too high to be acceptable. So then you have to go with 100% testing. For products in general, as we mentioned, the regulators working on the revised Annex 1 could not agree on a requirement for CCI testing on every batch, but they did agree that statistical CCI data needs to be generated. The industry has increasingly seen visual inspection, which is done 100%, and CCI inspection as being complementary. So defects that you pick up in visual will not necessarily be picked up in a leak detection process, and vice versa. In the visual inspection process, you will not pick up all leak defects. And again, this was specifically stated in this proposed Annex 1 revision. So the question then, becomes, if containers are already being handled for a visual inspection, does it make sense to include a concurring CCI inspection in this process? So that's a very logical thought process, and in fact, you have you see now, if you're familiar with inspection machine offerings in the pharmaceutical industry, that some suppliers have started to introduce what they call combination inspection machines. So these are machines that do both visual inspection plus leak detection. And a question that you could ask yourself is, will that approach meet Annex 1 requirements? And I would say it depends, and perhaps not necessarily. In this particular case, you will end up with a big complicated automated inspection machine. A lot of effort will be made to validate this, and it becomes part of your standard process, meaning that you lose some flexibility there. So for some facilities and for some products, this might not make sense. So what would be another option or other options to think about? So again, you need to think about, will CCI testing for you be part of a batch release, or is it more of a monitoring exercise versus some process studies that are done during scale up, for example? Would you prefer to have this heavier inspection process with automated combination machines, or do you want to leave yourself the flexibility to generate CCI data if and when needed? So, other options to think about here would actually be smaller, flexible inspection platforms that you could use to generate CCI data on a when needed basis. And the reason that why I'm saying this is, I think what you see out in the industry now are these combination final inspection machines, but I'll show you some examples which take this other approach. So what will be the impact on best practices for the industry in the area of CCI testing? Again, very often, blue dye and microbial ingress testing are the only CCI tests that are done during the product life cycle. I think that needs to change, and that will change. So it doesn't mean that you can't use blue dye and microbial ingress testing in certain situations, but if that's the only thing you're doing, I think it will be very difficult to meet future requirements, and in particular some of the requirements here in the proposed revised Annex 1. And that's mentioned here in the second bullet point, and even to follow some USP recommendations, current CCIT practices will need to evolve or change to prioritize the use of more robust analytical methods, which give you deeper insight into the product and process quality. And as mentioned before, regulators are being more critical in the areas of CCI method validation and the ability to detect critical leaks. And again, with these legacy methods, it's sometimes difficult to prove or demonstrate this. In the end, detailed best practices are evolving and are still not clear. And I mentioned this at the beginning of the presentation. I think there will be some period of time where there is a little bit of confusion and we don't have black and white recommendations or guidelines or guidances about this is exactly what you should do. But you also hear the regulators saying this. They do not want to be prescriptive. They don't want to give you a checklist. And from what I can tell, what they are expecting end users to do is to generate science-based data with arguments in the context of QRM to justify the CCI test methods and practices that are being implemented on a per product basis. So it brings it back to generating good data. So that was the first half of the presentation. What I'd like to do now is shift gears a little bit and look at a few industry case studies. These case studies describe very specific CCIT solutions, and what I hope this does is give you some concrete ideas, some concrete examples of things that could be done to meet these proposed EU Annex 1 revisions in the area of CCI testing. And to start off this section, just a couple of introductory slides. The first one actually from an FDA representative presentation given a few years ago. And you can see here, the very first slide, there is no gold standard CCI test. And I think sometimes in the industry, either the microbial ingress test or the dye ingress test is really seen as a gold standard. That's not true, at least from this FDA representative. That's not the case. The testing method should be appropriate for both the product and the final packaging configuration. And you can see here, again, a paragraph which really stresses the generation of science-based CCI data. So a test method is validated if it has been proven through scientifically accepted studies to be capable of detecting a breach in container closure system integrity. Test methods should demonstrate the ability to detect a container breach that is worst case or critical. So again, a little introduction there. So the blue dye or microbial ingress testing might not always enable you to do this. So what are some options? And for this, you can go to the USP chapter, and they have very nice descriptions of all of these technologies in this table. And in fact, this table was taken directly from the USP chapter. And you see on the left-hand side methods which they have identified as methods that can determine CCI in what they call a deterministic way, as opposed to the probabilistic methods on the right, which include the legacy methods of blue dye ingress or microbial ingress testing. And again, the chapter also specifically states that this is not an exhaustive table. There could be other methods that you could use. But what it does say is that these deterministic methods are preferred over probabilistic methods when other key method selection criteria permit. And of course, another point to look at is that some of these deterministic methods are non-destructive and can also be automated. And so those methods are then particularly interesting to enable statistical CCI generation for automated inspection in manufacturing or during scale up. So now let's look at a few industry case studies. This first one is titled Filling Line CCI Qualification. So this is an example where a manufacturer, in particular CMO, needed to qualify their filling line for CCI. As a CMO, you get in a lot of different packaging components, and you always want to demonstrate that your process is able to take the client's packaging components and produce good CCI. So in this particular study, you can see pictures of the equipment that was used here on the right. You see here a headspace analyzer, and you see here a CCIT vessel. This vessel is very similar in terms of purpose to the buckets that are used for the blue dye ingress test. So you can place samples in this vessel and condition them to force a change in the container, which will indicate a CCI failure with a headspace measurement. So what happens? The filling line produces empty sealed vials. So using their standard process, they will seal the packaging components. So basically stopper and cap and crimp the vials. The initial headspace is 1 atmosphere of air, which then contains 20.9% oxygen. You can include positive controls in this study. In this particular one, a 10 micron capillary was put through the stopper. And you can use gas ingress models to design an appropriate CCIT vessel cycle. And this is the advantage of deterministic methods. In some cases, because the method is quantitative and analytical, you can actually model it mathematically. And here, the cycle that is being talked about is pulling a vacuum while the samples are in here, and then injecting nitrogen as a backfill. And if you have a defective container, the nitrogen will ingress into these containers, and then of course the oxygen content inside the containers will change, and you can detect that as an indication of a leak. And so using the model here, you can see that a headspace gas flow model can be used to calculate the change in oxygen levels as a function of vial size, sample preparation cycle, and even an ideal defect size. And so you see here, you get an indication of what kind of oxygen levels you would measure for a particular container and a particular defect with the conditioning cycle that is chosen for the vessel. If you go to the data that was generated in this particular study, you see here that there are 2 different packages here. A 2R vial that was using a serum stopper or sealed with a serum stopper, the same vial sealed with a lyo stopper. And you can see that the samples that were produced by the process show no change in the original oxygen levels after being put through the conditioning cycle in the CCIT vessel. But if you look at the positive controls, they clearly show a drop in the oxygen levels, which was a result of the nitrogen in the CCIT vessel ingressing through the capillary defect and into the container. So this was a very straightforward way to generate filling line CCI qualification data. It's robust, science-based data based on analytical measurements. And just to give you a comparison, we've worked with a number of clients who use microbial ingress testing to qualify the CCI on filling lines, and you're basically go into a 2-month process before you have kind of formal certified results that you can present as a demonstration of good CCI on the filling line. A second case study involves 100% CCI inspection of a lyophilized drug product. And in this particular case, a manufacturer was producing freeze-dried product. They had a vacuum test as part of their batch release, and in fact, they started to see regular failures in the QC vials. And so the client decided to execute a process study. And here is one of those other options for generating statistical CCI data. We provided this particular customer with an automated lease inspection machine. So these machines are actually are small footprint, flexible. They can actually be delivered, installed, and qualified in a matter of weeks. And then at that point, you have a platform on site at the facility which can inspect and generate statistical CCI data. So inspect batches of product. This particular inspection machine was a headspace inspection machine, again, looking at oxygen content. Freeze-dried vials are typically packaged at an under pressure of nitrogen, so there should be no oxygen in the vial, and so a high oxygen content vial would indicate a leak. This particular client decided to inspect 6 consecutive batches, and what we're plotting here is the measured oxygen in every vial in those 6 batches. So in the end, it ended up being close to 180,000 vials. The batch size was roughly 30,000 vials per batch. And you can see here that there is clearly a problem. There are lots of vials with high oxygen content. You can see a fairly good batch here, but then you can see a fairly bad batch here, and you see everything else in between. This analytical data gives very deep insight into the product quality and what's going on with CCI, and clearly shows that this lyo sealing process is not robust. So these 6 batches, it was the same packaging components, the same product, but somehow the sealing was not robust. And we've looked at this more carefully, especially with freeze-dried product. Here is a plot of the reject rate per quarter of a 100% leak detection machine installed to inspect freeze-dried product at a particular facility. This is 4 years of manufacturing data, so 156 lots, almost 2 million vials. And what you see from this data when you analyze it is that 44 lots, or 28%, had 0 rejects. So those were batches that did not have CCI failures. 3 lots had a greater than a 2% reject rate, and the average reject rate was 0.27%. And you remember talking about the ampule sealing process, which has inherent CCI risk. Typically, ampule lines are probably rejecting on the order of 0.1%, 0.5% of product, and that's roughly what was seen here for this freeze-dried product. So we've actually looked at data from more than 17 million commercial vials, freeze-dried vials from global facilities, and it does show that it is difficult to manufacture a perfect CCI lyo batch. And I think when you think about the sealing process, it's understandable in the lyo process as opposed to a normal or standard liquid filling process. You are using the lyo shelves to seal the vials. You know, those shelves are compressed. You have thousands of vials on the shelves, and the compression of the shelves inserts the stopper into the vial. And then these vials are unloaded from the freeze dryer, and it can take sometimes up to 24 hours to actually get all of the vials to the capping and crimping machine to be capped and crimped. So clearly there's room there for risk to CCI. So again, this is an example of where statistical data allows you to determine a fundamental baseline failure rate. You need to go back and put in process controls, and then ideally you would then start remeasuring to see if your process controls improve the situation and was able to eliminate enough of that risk to be acceptable. The final case study here has to do with a raised stopper study, again for freeze-dried product, because here, as just mentioned, your freeze-dried product can sometimes, it will at least take hours and sometimes up to a day to be capped and crimped. And during that time, there is risk that this stopper that has been pushed into the vial will start to move its way up or has not been properly sealed or pushed into the vial. In this particular case, the vial was a 2R vial, and you can see the description of the packaging components there. A pilot scale freeze dryer was used to prepare empty vials which had shims. And so you can see a photo here of a shim between the stopper and the vial neck. These shims had different thicknesses, and there were 400 of each type, so 1,600 vials in total. Again, an example for doing a study with statistical sample sets. You can see that the vials were backfilled with a slight underpressure, and then the vials were removed from the chamber with shims in place and stored in ambient air, tested at multiple time points. And again, with an underpressure of nitrogen inside the vial, a headspace oxygen measurement was a very useful analytical tool to detect raised stopper samples that were beginning to leak. And so these vials were analyzed, were pulled at different time points for this particular study. And if you look at some of the results from this study, in this case, you can see that leaks started to happen in these vials at the 1 and 1.5 millimeter shims. You started to see a few leaking vials due to these raised stoppers. And by the time that you hit a stopper height of 2 millimeters, you had a failure rate of 21%. And these are, I think, the types of studies that the regulator will want to see when they say that your raised stopper height needs to be qualified. So your stopper height sensor needs to be set at a certain limit, which needs to be qualified. And just picking 1 millimeter will not be sufficient. And we've done a number of these studies, and the probability of losing CCI can be definitely a function of the very specific packaging components that you're using. So I think you will possibly see a range of different stopper heights that will be implemented on the stopper height sensor depending on the package that is being used. Very quickly, some extra data that can be collected here. The client was interested in do these raised stoppers also involve the ingress of moisture into the vial. Obviously, if it's a freeze-dried product, moisture is a stability indicator. So here, the CCI issue is not only related to sterility, but also there's a stability consideration. And you can see here that water vapor levels in the vials, if the stoppers were raised, definitely showed an increase over time. And studies have also shown that this water vapor that gets into a leaking vial will eventually make its way into the product, and therefore will be a stability risk for the sample. I wanted to end this presentation with a thought experiment. Now, at the very beginning, it was mentioned that my background is atomic and molecular physics. So during my study times, I studied a lot of, or all of, Einstein's thought experiments. Einstein, in German, you call them Gedanken experiments. Einstein was a theoretical physicist, so he did all of his experiments in his head, and that's how he figured out a lot of things. And I thought, I've done this now a few times, and people have found it useful to do a thought experiment about CCI control strategy. You can perhaps try this with your teams and see if this gets good discussion going. So think about, and then here's the thought experiment. Think about the CCI control or testing strategy currently implemented in your company. And you might not be directly involved, and actually you might not know the details of it, but you can probably guess or have some idea of what this strategy is like. Okay, so if you've got that in your mind, going back to this case study that we went over, if your lyo sealing process is doing this, would you know about it? So you can think about that. After you've thought about that question, the next question would be, when would you know about it? Would it be after one batch? Would it be after 6 batches, as shown here? Would it be after 30 batches? And then finally, and perhaps the most important question, what would you need to do to prevent this from happening? So again, some very straightforward, easy questions, but if you walk through those, I think it will raise some points of discussion in your own facility, in your own CCI strategy about what you perhaps maybe should be or should not be doing. So from final thoughts, the final slide, meeting the proposed EU Annex 1 requirements, use an analytical CCI test method when possible. Again, that's basically one of the things recommended from USB 1207. Especially if you're going to generate statistical CCI data. The legacy methods will not enable you to do that very well. More CCI testing data needs to be generated. Again, using statistical sample sets or full batches, as the case study showed, you gain real science-based insight into process and product quality, which in turn will support decisions about your control and testing strategy. And finally, perhaps a little bit of a provocative question, and this relates to doing visual inspection. Again, this revised EU Annex 1 version strictly says just a visual inspection alone is not considered sufficient. If you look at the effort and the resource that goes into particle contamination control, considering the risk to the patient, should there at least be equal effort to detect particles and to detect CCI defects when compared to the effort used for detecting particles. So again, here at Lighthouse, we do support our customers with a lot of CCI activities, and looking at these new regulations that are coming out, what we really want to do is provide solutions that will enable manufacturers of sterile product to qualify their CCI, develop the appropriate test methods, and to do these studies which give insight into product and process quality. And I hope this presentation has given you ideas. Thank you very much for your attention. And we will now move into what I think will be a short Q&A session, because we're just about to the end of this time, or the end of our time here. Hopefully some of you who have submitted questions with the chat function have already gotten answers. I just have a few questions here that have come in, which I will attempt to answer. The first question here that is on this paper here that I have: define CCIT during the entire product life cycle. Did the EU drafters understand the difference between? Oh, sorry, that's a separate question. So the entire product life cycle. So what I mean there is going back to development. When you're doing packaging development and trying to pick your packaging components, have you generated good CCI data, which really shows that this package is fundamentally a good package for your product? In manufacturing or during scale up, have you been generating some CCI data there to show that the process has not had any impact on the fundamental ability of your product to maintain closure? And then finally, are there stability studies being done which generate CCI data over shelf life? So long term, does this package maintain good CCI? All right, another question here. Did the EU drafters understand the difference between AQL testing and regular statistical sampling, or was the emphasis for production only and not other packaging validation studies? A very good question, and I think that's one that comes up a lot. I don't know the answer to that because I was not part of their discussion, and I don't know the draftees exactly. I haven't heard them mention it, and I think this will be, some area where people will have to make some good decisions about. Of course, the EU Annex 1 is applicable to the manufacturing environment, and I know that visual inspection uses a lot of AQL approaches there. I think it is a different scenario when you talk about other packaging validation studies, and I think there the comment would be, don't just take 10 or 20 samples when you're doing a packaging validation study. You're more likely, wanting to look at 50 or 100, or in the race stopper study that I presented here, hundreds of samples. Let me do one last question. I see our time is up, but I'll get to this one here. I am curious what you believe the expectations will be for early phase products. You won't have process controls in place for phase 1 and phase 2. Again, I think it goes back to this product lifecycle approach, which I would actually recommend you reading the USP 1207 chapter if you have not done that already. It's 50 pages, but it is very well written and very easy to read, and it does talk about generating data, early phase, and I think there what you want to do is just start doing CCI testing to understand the package and CCI, and I think that's where you start building up what's called a database of CCI data, as the USP 1207 calls it. And I think what is a challenge is bringing that data through the whole process, so that by the time you hit commercial manufacturing, that the people who are actually manufacturing the product are familiar with what has been done in early phase and can take appropriate actions for the process controls perhaps depending on the insight of early CCI data that has been generated. Okay, again, we're past time now, so we will end here. Again, thank you very much for your attention. Other questions we're happy to receive with follow-up. Don't hesitate to contact us. And as Marjolein described before, a recorded version of this webinar will be made available. Thank you very much and have a nice day or nice evening.